Hello students, in previous lecture we have already discussed about nucleic acid, component of nucleic acid, structure and functions of the DNA and RNA. Today we will discuss about DNA replication and DNA replication by semi-conservative method. So, DNA replication and duplication. Duplication means the formation of the new molecules, same molecules. DNA replications means replicate the DNA and synthesize the same DNA copies. Means the DNA is the genetic material. When cells divides, the daughter cells receive an identical copy of the genetic information from the parental cell. The replication is a process in which DNA copy itself to produce identical daughter molecules. It is a complex process and involves a series of steps. Means replications, the DNA molecules, they can synthesize the self new two DNA strand, because DNA is double helicals, they can synthesize new DNA strands. Means parent strands, they can synthesize the new offspring strands or the formation of the new strands. In case of the replication in semi-conservative methods, the parental DNA has two strands, both are the complementary to each other. They undergo simultaneously replication to produce the two daughter molecules. Each one of the newly synthesized DNA has one half of the parental DNA and one half of the new DNA, and this type of replication is known as the semi-conservative. Since means parental DNA molecules, the one stands, there is the synthesis the new DNA molecules, it is the daughter strands and both are the complementary to each other. There is the half strand, it is the parental, and one half strand, it is the synthesis, it is the newly strand. That new strand, it is the offspring or it is the daughter strand. That type of the process, it is known as the semi-conservative replication process. Firstly, this experiment was discovered, or the evidence of this experiment, there is the Michelson and Stark in 1958. Now, how to exactly start the replication process? Means there is the initiation of the replication. The initiation of the replication, the DNA synthesis occurs at the site. It's called as the origin of replication. Means where is the process is the properly occur, the synthesis of the DNA molecule. That site, it is known as the origin site. In case of the prokaryotics, it is the single site, whereas in eukaryotics there is the multiple sites. These sites, it's contain the mostly the consist of these sites, there is the short sequence of the adenine and thymine base pairs. The specific protein molecules DNA, means there is the 20 to 50 monomers of the nucleotides, bind the site of the origin replication, and this cause there is the development of the double-stranded DNA to separated. Means DNA strands both are the separated, means there is the development of the bubble-like structure. These bubbles develop means just like in prokaryotic there is the single origin sites, means there is the development of the single bubble. In case of the eukaryote there is the multiple sites of origin, means there is the development of the numbers of the bubble. Okay. These two complementary strands of the DNA molecules, they are the separate. The separated strand, there is the formation of the replication bubble. See in the diagram, the DNA double helicals form. There is the binding of the protein molecules. That DNA protein molecules, there is the binding to the presence of the adenine and thymine base pairs or the origin site, and there is the development of the bubbles. There is the single strand DNA template ready for the synthesis of DNA. Now, actually, what happened into the replication process basically? See in the diagram, there is the DNA replications, there is the formation of the bubbles because there is the adenine thymine base pairs and DNA protein molecule binds. There is the separation of the two strands. That's why there is the formation of the bubbles. According to the bubbles, the both strands they are separated. These separated two strands, there is the development of the leading strand and lagging strand. Now, what is the leading and lagging? The meaning of these words: leading means the strand they continuously lead or the continuously synthesize. Lagging means they can synthesize but not the continuous in manner. Okay. Then formation of the RNA molecules with the help of the DNA polymerase third, DNA polymerase first. Second, there is the need of this process, the synthesis process, RNA primer. For the synthesis of new DNA strand, the short fragment of the RNA require as a primer. Primer means there is the existing near about the 5 to 15 base pairs. These binds to the DNA template strand. These both are the complementary to each other, and they supply the primers the continuously on the lagging strand of the DNA. But in contrast to the leading strands, is almost the single RNA primer is the necessary. Now, means there is the lagging and leading strands. The both strands they are the run into the opposite direction or both are the anti-parallel to the each other. The DNA synthesis process, it is the semi-discontinuous and bidirectional method. Semi-discontinuous and bidirectional means basically in replication, one strand run into the 5' to 3' direction simultaneously on the both the strands of DNA, and one strand is the leading strand of the DNA synthesis, it is the continuous in form, and lagging strands, it is the DNA synthesis, it is the discontinuous in form. Now, in this process, basically why the formation of the lagging and leading form? Actually, this is the basic problem of the polarity. Means, in case of the DNA strands or the leading strands, there is the free 3' hydroxyl group. So, there is the replication frog, the replication strands sequentially addition of the new nucleotides continuously. But in lagging strand, it's run into the 5' end and presents some problems. So, there is no DNA polymerase enzyme. There is the catalysis, the addition of the nucleotides at the 5' end in direction and growing the chain, but that chain they are not the continuously synthesized, they can synthesize in the small fragment, it is the lagging strand. Now this process, so that's why there is the replication synthesis process occurs in the direction and bidirection forms from the origin of origin points. Next, replication frog and DNA synthesis. Actually, the separation of the two DNA strands of the parental DNA results in the formation of the replication frog. The replication frog move along the parent DNA and the daughter DNA molecules are synthesized. Now, basically, there is with the help of these replication bubbles formation, DNA strands stabilizing, there is development of the bubbles, RNA primers binds to the one end 5' to 3' and second is the 3' to 5'. One is the run towards the replication frog and one is the away the replication frog. This away from the replication frog formation, they are the stabilized with the help of the DNA helicates. These enzymes binds to the both DNA strand at the replication frog. So helicase move along to the DNA helix and separate the strand. The functions of these helicates basically depends upon the just like the zip opener, or in case the functions or the helicase depends on the ATP energy for supply the ATP energy. Now second thing is that the single stranded DNA binding protein. The DNA helix stabilized protein, it is the SSB protein, binds only the single DNA strand or the separated by the helicates, means there is the development of the frog helicase apply, there is the formation of the frog. After that, the both strands they are the stabilized. It is not the enzyme molecule, it is the protein molecule. They could not be destabilize the DNA segment. The both strands they are the separated and provide the template for the newly synthesized DNA molecule. Second and most important, it is the DNA synthesis process. There is a requirement of the enzyme, it is the DNA polymerase third. Now, what is the basic function of the DNA polymerase third? The DNA polymerase third 5' to 3' direction, and this is the anti-parallel to the parental templates DNA strand. There is the addition of the 4 nucleotide deoxyribose nucleotides, means there is the synthesis of the DNA, means DNA it is the made up from the basic 4 bases adenine, guanine, cytosine and thymine. These 4 nucleotides is the important. So that's why there is the DNA polymerase third one by one adding the nucleotides triphosphate. There is the releasing of the growing the chain into the 3' end and releasing the pyrophosphate molecules. Now in this way the chain is the elongated to the parental strand. Newly synthesized DNA strands they are the complementary, and simultaneously in case of the another second strand, there is the that strand, it is a direction from the 5' to 3' direction towards the replication frog. Towards the replication frogs, that stands, there is the replication which is the continuous. Okay, and now this continuously from there is not the need to the addition of the any type of the primer, they are the continuously synthesized. But the another direction 5' to 3' direction, in this direction it is the away from the replication frog, so there is the discontinuous in form due to the polarity problem. And next is the formation of the Okazaki pieces, because the one strand leading strands they are the continuously synthesized strand, or the lagging strands it is the away from the replication frog but they are the discontinuous in manner synthesized. This discontinuous manners there is the formation of the small species of the newly synthesized DNA molecules, they complement to the parental strand, but there is the formation of the small fragment. That fragments is nothing but it is the Okazaki fragments. With the help of the DNA polymerase first, these strands again with the help of the ligase enzymes, it is the responsibility of the DNA polymerase first, and they can join the such types of the strand and complement it to the parental strand. Means discontinuous strands they are again continuous, this lagging strand to the parental DNA molecules, and both are the complementary to each other. Another one function of the DNA polymerase third, it is the proofreading function. Proofreading function means addition of the one by one nucleotides, they can check the same or the complementary base pair added into the parental strands, newly synthesized strands, and these strands is the complementary to the parental strand. It is the basic function, another one function of the DNA polymerase third. Next is the replacement of the RNA primer by DNA. Actually newly synthesized strand continuously till it is closely proximate to the RNA primer. Now the DNA polymerase come into the picture. It remove the RNA primer and take its position. DNA polymerase first catalyze the synthesis of the 5' to 3' direction, and the fragment of DNA replace RNA primer. Now this with the help of the enzyme ligase, the phosphodiester linkage between the DNA synthesized by the DNA polymerase third and small fragments of the DNA producing DNA polymerase first, with the help of the next sealing they require the energy and you can provide the breakdown of the ATP to AMP and pyrophosphate. Means there is the discontinuous form or the small fragments, they are the sealing, as well as continuously synthesized both strands they are the complementary to each other, so there is not the need of the primer molecule. That position takes the DNA and come into the picture of the DNA polymerase first. After that, the synthesis of the two strands, they are the synthesis of the complementary of the two strands, they are the after the formation of the supercoils. The supercoils means what happens, suppose for example in case of the ropes, at the one end they can bind and stretch the ropes. If they are the again release the tension, what happens, there is the formation of the coils. Same way that in case of the DNA molecule, they are the continuously synthesized, but it is a single strand. So that's why there is the formation of the coils. With the help of these topoisomerase, there is the two types of these topoisomerase molecules. These topoisomerase, they can DNA strands overcome the problem of the coils. They release the strand firstly, and second receive this strand, and second types DNA topoisomerase or this gyrase cut the strands and release and overcome the problem of the supercoils. Now this is the function of the topoisomerase. And involvement of the enzymes in DNA replication process. There is the DNA polymerase first, second, third, or the helicase. DNA polymerase first replace the primer molecules. DNA polymerase second, repairing the functions. DNA polymerase third, addition of the nucleotides. And helicase, they can stabilize the both strands. There is the formation of the replication frog. There is the formation of stabilize the with the help of the helicase. Then ligase sealing the Okazaki fragments, primase for the need to the replication or the start the replication, and last is the topoisomerase, the formation of these coils or the unwinding breaking of the molecules. They are the separated or the formation of means release the tension on the DNA molecules. And thank you.